DNA sequencing strategies, dideoxy sequencing or the Sanger's method. So-called first generation sequencing technologies which emerged in 1970s included the Maxam-Gilbert method, which was developed by Alan M. Maxam and Walter Gilbert, and the Sanger method or dideoxy method, which was developed by Frederick Sanger. In the Sanger method, which became the more commonly employed of the two approaches, DNA chains were synthesized on a template strand, but chain growth was stopped when one of the four possible dideoxynucleotides which lack a 3' hydroxyl group became incorporated, thereby preventing the addition of another nucleotide. A population truncated DNA molecules was produced that represented each of the sites of the particular nucleotide in the template DNA. The molecules were separated according to the size by electrophoresis, and the inferred nucleotide sequences was deduced by a computer. Later, the method was performed by using automated sequencing machines in which the truncated DNA molecules labeled with fluorescent tags were separated by size within thin glass capillaries and detected by laser excitation. The process of DNA synthesis and replication involves DNA helicase, DNA polymerase, DNA template and deoxynucleotides. DNA replication starts when DNA helicase unravels the double helix structure to a single stranded DNA and form a replication fork. RNA primase introduces a primer that binds to the single stranded DNA. DNA polymerase then binds to the replication fork and starts DNA synthesis by sequentially adding nucleotides to the 3' hydroxyl end of the RNA primer bound to the DNA template. The result is the creation of an extension product. The DNA sequence is copied with high fidelity because at each base on the DNA template, DNA polymerase incorporates the nucleotide that is complementary to that base. Thymine is complementary to adenine and guanine is complementary to cytosine because they can form hydrogen bonds with each other. Sequencing of the model genomes is most important application of sequencing in the last 2 decades. It consists of two basic steps. First, many copies of subsequences of a few hundred of base pairs or BP are generated by the sequencers. These small pieces of sequences with every base known are called as reads. Then the reads are assembled to reconstruct the genome. There are possible base calling errors in the read generation step because efficient methods or algorithms are needed to analyze image data and correctly call a base at each position. At the assembly step, there is no prior knowledge of how the reads are supposed to fit together, and furthermore, assembly is analogous to solving a massive one-dimensional jigsaw puzzle with the pathological property that many of the pieces occur many times in different places due to the repetitive nature of genomes.